What is the goal of your experiment?
The goal of our experiment is to see if we can design a strain of chitinase that will effectively degrade chitin, and also if it can be evolved using enzyme evolution and error-prone PCR.
What is your construct? Why did you choose the genes or strains that you did?
So our construct is basically the gene insert into the vector, and we chose Serratia marcescens strain of chitinase because it's been proven in previous studies to be one of the most efficient and impacts the most species of fungi. The GST strain was used so that we can use it for protein purification, and the flag tag was used so that when we do Western blotting, we can use an anti-flag antibody.
What are the basic steps of the experiment?
So first we design our construct, and then we insert the gene of interest into the PBAD D4 vector, and then after that we check expression, and then we plate those cells to see if the cultures are actually viable. Then we purify the DNA, test for protein expression, and also we did an arabinose induction check to see what concentration we need to maximize induction efficiency. And then finally, we purify protein, check if there's protein at all. Then we'll do error-prone PCR to see if mutagenesis is possible.
Can you please explain the purpose of your chitin assays? How do they work?
So the chitin assays are kind of like a test to see if the chitinase strain that we have chosen is working. The solution assay uses horseradish peroxidase, and the chitinase degrading the chitin in the solution will react with the HRP or horseradish peroxidase and will emit a light, so that we can measure that light and measure the concentration of chitin that's left in solution to test degradation. The chitin agar plate solution is basically we just plate colonies with our gene of interest and see how much chitin degradation there is on the plate.
How do you use Western blot in this experiment?
So the way Western blot works is you get primary antibodies that recognize your construct, which is why our construct has the flag tag gene in it, so that when we do Western blot, the anti-flag antibody can recognize our gene of interest. And then a secondary antibody that has a HRP strain protein attached to it will recognize the primary antibody, the anti-flag, and then we can react it with luminol in this case to produce light. And then we use film to capture that light and see how much protein we actually have.
What are the impacts of your project and how could it benefit society?
So chitin is a very common polysaccharide in nature, but the reason why we're trying to see if we can degrade chitin is because it's a major component of fungal cell wall. Serratia marcescens chitinase specifically impacts many different species of fungi, but the main goal is to see if we can design a construct where we can degrade chitin so that we can use chitinase to degrade the fungal cell wall of species such as Bipolarus sp that impact wide range of agricultural products, and thus we can reduce the cost that results from fungal infection.
What kind of challenges did you face during the experimentation process?
So there were a lot of redos, as a lot of times the experiment wouldn't work very well, the induction efficiency wouldn't work very well, so we'd have to tweak procedures here and there to try and increase the efficiency. PCR, it took a really long time to get the optimal PCR conditions. It's a lot of trial and error, but eventually we were able to optimize all of our conditions.
What can you conclude based on the result of the experiment?
We can conclude that our strain of chitinase can be mutated, and since mutagenesis is possible, then it's also possible to evolve the enzyme to make it more efficient. We can also conclude that our strain of chitinase can degrade chitin.